Meet Sir Hans Adolf Krebs, the brilliant architect of metabolism. This presentation explores how he unlocked the foundational, cyclical engines of life itself. We will journey through his groundbreaking discovery of the urea cycle, the body's primary method for removing toxic ammonia, and delve into the intricate mechanics of cellular respiration. By mapping out what we now universally celebrate as the Krebs cycle, he illustrated how cells transform nutrients into the vital energy that sustains us. From citrate to oxaloacetate, get ready to decode the elegant, blueprint-like pathways of biochemistry that define our very existence. Now let's decode the engine of life itself by looking at the monumental impact of Krebs' discoveries. First, he triggered a massive paradigm shift in 1932 by discovering the first metabolic cycle in biological history, proving that cellular chemistry relies on regenerative loops rather than linear decay. To understand the scale of this engine, consider this. The average human turns over a staggering 40 kilograms of ADP, our fundamental cellular energy, every single day using his namesake Krebs cycle. Finally, we look at the recognition. For fundamentally altering our understanding of biology, Hans Krebs was rightfully awarded the 1953 Nobel Prize in Physiology or Medicine, an honor he shared with Fritz Lippmann. Let's look at how the foundations of this great biochemist were built between 1900 and 1925. It began with the discipline instilled during a strict, Spartan upbringing in Hildesheim. Intense Sunday botanical walks with his domineering surgeon father forged a rigorous work ethic and razor-sharp observational precision. Then came the spark of his academic journey. Moving from the University of Göttingen to the University of Freiburg in 1919, he was profoundly influenced by Franz Knoop. Knoop taught Krebs that intracellular chemistry operates through highly organized pathways mapped via logical, chemical tracing. This vital lesson accompanied Krebs throughout his time at the University of Munich in 1921 and the University of Berlin in 1924, shaping his future discoveries. Every master needs a toolkit, and from 1926 to 1930, Krebs underwent a transformative apprenticeship in Dahlem. Here we see the anatomy of his experimentation. It began with the mentor, the dictatorial yet brilliant Otto Warburg, under whom Krebs worked at the Kaiser Wilhelm Institute. Warburg's laboratory provided the cutting-edge techniques required to isolate cellular activity. First, the tissue slice technique involved cutting tissue to a thickness of just 10 cell layers, allowing oxygen and carbon dioxide to diffuse freely. Second, the manometer enabled the incredibly precise measurement of oxygen consumption in this living tissue outside a whole animal. Armed with these precise methods, Krebs was ready to crack open the secrets of metabolism. In 1932, Krebs faced his first major challenge, Puzzle 1, the ammonia problem. The core of the problem lay in protein metabolism. Amino acid breakdown creates highly toxic ammonia. How exactly does the mammalian liver detoxify it? To find out, the test began. Krebs and medical student Kurt Henseleit systematically tested different chemical compounds on live liver slices. This led directly to the breakthrough. They discovered that adding ornithine and citrulline massively accelerated urea production. Crucially, the citrulline was not consumed by the reaction. Instead, it acted as a continuous catalyst. This served as the hidden catalyst key, proving that the liver funneled toxic waste into a safe, excretable form via a repeating, circular pathway. This brings us to the formal discovery of the urea cycle, truly biology's first metabolic wheel. The core realization here represents the paradigm shift. Before this moment, scientists assumed metabolism was merely a linear chain of decay, working strictly from point A to B to C, and straight to waste. Krebs completely upended that view. He realized that cellular chemistry relies on a regenerative wheel. By feeding toxic ammonia and carbon dioxide into the loop, Intermediates such as citrulline and arginine flow through the pathway to spin out harmless, excretable urea. Crucially, the foundational intermediate, ornithine, is continuously regenerated at the end of the loop, keeping biology's engine spinning indefinitely. History takes a dramatic turn in 1933 with a story of exile and resilience. First comes the dismissal. In April 1933, despite his brilliant work on the urea cycle, Krebs was fired from his university post in Freiburg under Nazi anti-Semitic laws. But then came the rescue. Sir Frederick Gowland Hopkins invited Krebs to Cambridge, England, a crucial move funded by the Rockefeller Foundation. Finally, let's look at the luggage. While fleeing Germany, Krebs managed to bring 30 of his highly specialized Warburg manometers. It was a cargo far more valuable to his future, and the future of science, than the Nazis ever realized, ensuring his revolutionary research could continue uninterrupted in exile. Settled in England, Krebs confronted Puzzle 2, the black box of respiration. We begin with the baseline, using the pigeon breast muscle tissue as our control. It consumed 670 micromoles of oxygen. Next, look at the spikes. When Krebs introduced 4 carbon compounds, succinate, fumarate, and malate, oxygen consumption didn't just rise. It spiked disproportionately, more than doubling the baseline. This led to the deduction. Because oxygen consumption increased far more than required to simply burn the added substances, they couldn't be just fuel. Instead, they had to be acting as continuous catalysts in a cyclical reaction. The black box of cellular breathing was beginning to open, once again pointing to a magnificent metabolic wheel. In 1937, working alongside William Arthur Johnson in Sheffield, Krebs finally solved the grand puzzle, the citric acid cycle. He brilliantly connected carbohydrate breakdown to the organized pathways he had learned from Franz Knupp years prior. This elegant blueprint reveals the universal engine that powers oxygen-respiring life. At its core, a 2-carbon acetyl fragment derived from food combines with 4-carbon oxaloacetate to forge 6-carbon citric acid. As this magnificent molecular wheel turns, carbon dioxide is exhaled, and vital high-energy electrons are extracted to generate ADP. It is the ultimate metabolic loop, converting what we eat into the very spark of biological life. Let's look at a comforting reminder that even the greatest triumphs face setbacks. The most famous rejection in science. In June 1937, Krebs submitted his monumental discovery of the citric acid cycle to the prestigious journal Nature. In an astonishing historical blunder, Nature rejected the manuscript, politely noting they had sufficient letters to fill correspondence columns. Undeterred, Krebs published his breakthrough in enzymologia instead. Fast forward to 1953, an ultimate vindication arrived. That very same rejected discovery earned Sir Hans Krebs the Nobel Prize in Physiology or Medicine. It stands as history's ultimate proof that true genius cannot be sidelined by a rejection letter. Even after winning the Nobel Prize, Krebs continued to untangle cellular mysteries, leading to Puzzle 3, the glyoxylate bypass in 1957. Consider the problem. How do plants and bacteria manage to survive solely on a simple 2-carbon acetate? It seemed impossible since the standard Krebs cycle exhales carbon dioxide and loses carbon atoms at every single turn. Here lies the solution. Working with Hans Kornberg, Krebs discovered an elegant metabolic shortcut. 2 specialized enzymes, isocitrate lyase and malate synthase, slice straight across the standard loop. By bypassing the carbon dioxide-losing steps entirely, this clever detour allows organisms to conserve carbon, enabling the net synthesis of larger, complex molecules from nothing but simple acetate. To conclude our masterclass on Sir Hans Krebs, let's synthesize his life's work through the Krebs Trinity, a diagnostic matrix. This comparative breakdown maps his 3 monumental discoveries. First, the urea cycle, discovered in 1932, serves to detoxify ammonia in mammals and amphibians, relying on citrulline as its catalyst key. Second, his crowning 1937 achievement, the citric acid cycle, which extracts vital ATP energy across all oxygen-respiring life, driven by oxaloacetate. Finally, the glyoxylate cycle of 1957 allows plants, fungi, and bacteria to synthesize biomass directly from simple acetate, utilizing isocitrate lyase as its clever enzymatic shortcut. Together, these three wheels form the foundational trinity of modern metabolic biology. During the Oxford years, pacemakers and control, from 1954 to 1967, Krebs moved beyond simply mapping metabolic pathways to figuring out how cellular traffic is actively directed. He focused his brilliant mind on gluconeogenesis, the complex process of creating vital glucose from non-carbohydrate sources. In doing so, he identified crucial pacemaker enzymes. Operating like strict, one-way valves in a plumbing network, these enzymes use feedback loops to regulate metabolic flow. By controlling the pace and direction of reactions, they act as cellular traffic cops, successfully preventing the cell from wasting precious resources and burning out in futile, continuous loops. Let's reflect on the ultimate synthesis, the cyclical paradigm. Before Krebs, the scientific consensus viewed metabolism as a primitive, linear conveyor belt in which food simply turned into energy and then ran straight to waste. Krebs completely revolutionized this view with the concept of cyclical regeneration, beautifully illustrated here by interlocking gears turning in an infinite loop. Why did evolution choose cycles? Because circular pathways are nature's most efficient mechanism. They allow cells to continuously reuse catalytic intermediates, squeezing out maximum ADP energy from minimal raw materials. This repeating design is the elegant, sustainable blueprint behind all complex life. In closing, let's reflect on the legacy of the loops. Sir Hans Krebs fundamentally transformed our understanding of life by replacing the old, linear model of metabolism with the beauty of cyclical chemistry. His work proved that the biological world doesn't simply consume and discard. Instead, it relies on beautifully engineered, regenerative wheels that recycle components to sustain life efficiently. By discovering the urea cycle, the citric acid cycle, and the glyoxylate bypass, Krebs mapped the very machinery that keeps every cell breathing, powering, and renewing itself. His monumental discoveries endure as the foundational, spinning gears of modern biochemistry, forever illuminating the elegant, circular rhythms that define all living things.